Thank you, Professor Chen, for the very kind introduction. I'm really honored to give this lecture, the Yuen Kwok-yung Lecture in Life Science. And one interesting reason why I accept the invitation is because my name, if you look at it, is actually with 10 out of 12 alphabet letters, which are almost the same as the Yuen Kwok-yung Kwok-yung. And that is an interesting reason why I should accept the lecture. But there are other important reasons which the chairman may not know. In 1984, 5, and 6, I was honorary clinical assistant professor of the Department of Surgery of CUHK when they started the medical school, initially in United Christian Hospital, when I was a medical officer there. And another reason is because in 2003, Professor Joseph Song invited me to see the first SARS patients in Prince of Wales Hospital. And then he appointed me as your honorary professor in medicine and therapeutics for 2 years in 2004 and 5. And therefore, I have every reason to come here to give this lecture on preparation for the next pandemic due to an unknown disease X, the Hong Kong perspective. Now, so this is the timeline of the pandemics. You can see here that we basically put it down from many years before, 541 to 2023. And you can see that initially, before the 19th century, it's mainly due to bacteria, like plague and cholera. Now, but these are all controlled with better sanitation, good water supply, separate sewage system, and also antibiotics and vaccines. Now, since then, starting from 1918, we have influenza and then coronaviruses as causes of pandemic. And there are many emerging infectious disease outbreaks, some of which almost become a pandemic like SARS 2003. And you can see that these are no longer bacteria. They are viruses, and especially RNA viruses, which are spread by the respiratory route. And that is very important because RNA viruses tend to mutate much more rapidly than DNA viruses or bacteria, fungus, and parasites. And there is every reason why they should become the dominant causes of outbreaks and pandemics. And worst, of course, is they spread by respiratory droplets and aerosol, which is very difficult to control. Now, of course, they are causing a lot of mortality. So if you look at 1918, what we call the Spanish flu H1N1 pandemic, there is an estimation that at least 17 million people died. Some say 100 million, so I took something in the middle, around 50 million. And you can see that every time you have a pandemic, you have around at least 2 million people who died within a short period. And in 2018 COVID now, the counting is around 7 million, but the actual number should be much higher. And of course, because of all these findings, our prediction for the next pandemic due to pathogen X or disease X would be very likely another respiratory RNA virus. Now, so why should we have all these emerging infections, all these pandemics coming? And we have to understand it from what we call One Health perspective. So One Health, we are looking at the human health, animal health, and the health of the ecosystem that we are all living. Both the animal and the human are living in the same ecosystem. Now remember microbes, the genome, all the genes adding together would determine the phenomes, the express characteristics of these microbes. But actually when the genome and the phenome change by genetic mutations, recombination, or reassortment, that is 洗牌, the reshuffling of the gene segments, for example, they all do this because they try to adapt to the changes of human behavior and also the changes in the ecosystem. And remember, the most important cause of changes in ecosystem is because of human behavior and what are happening in the human population. We have rising number of people, aging population, which increase the susceptibility to infection. We have socio-economic expansion, which infringe on wildlife habitats and the demand for meat proteins. And then we have changing human behavior, such as sexual practices, game food, abuse antibiotics, etc. And also, very importantly, global interconnectivity, especially air travel. And all this, of course, will lead to changes in the ecosystem like climatic changes, extremes of temperature, collapse of non-human habitats, and also increase in the arthropod range. So you have much more mosquitoes nowadays, expanding to the north and also towards the south because of global warming. Unfortunately, all these changes are not matched by our hygiene, public health, and regulatory measures. They are all lagging behind, especially in the farm market, abattoir, and also our sanitary systems. And that, of course, fuel the emerging infection, re-emerging infection, and then a potential pandemic agent will start to become mature and then cause the next pandemic. Now, so how can we tackle pandemic problems? How do we tackle emerging infectious disease problem? The simplest way to do it is to understand the total microbiome. So every microbial species in the world, if we do surveillance, if we get all the microbial genome sequence and understand their expressed characteristics, that is their phenome, then we should be able to prepare for the next pandemic very easily by making diagnostic tests ready, antimicrobial agents, and of course, vaccines. With all this available, we have no fear for the next pandemic. If we know the total virum, bacterium, fungium, parasitum, then we have no fear. But look at the total number of microbes on plants and animals. The number is staggering. We are talking about 1 + 18 zeros behind, just for non-human animals. And of course, the mammals is equally worse, and then birds, bats, camels, mosquitoes, and trees. And how about the number of microbiomes in the environment like the soil, the ocean, and atmosphere? And as you can see, this is an astronomical number of virus species. Just look at virus. Don't ignore about the bacteria, fungi, and parasites. And more importantly, the diversity of these viruses evolve rapidly with time. And so this is almost basically an impossible task, trying to tackle pandemic by understanding of the total microbiome using surveillance. Now, but we have other understanding. We know that emerging pathogens, 75% of them are coming from animals, and they are the candidates for the next pandemic. Now, so the lessons, especially in Hong Kong, is that the bats, from the bat viruses, it comes the SARS-CoV-1, 2, the MERS-CoV, which is the Middle East Respiratory Syndrome coronavirus. And also from the wild birds, we have the avian flu, H5N1, H7N9, and others, many other, X and NY. And these actually come from wild animals to start with, which has a high biodiversity. You have many 3,000 bat species, for example. They jump into the civet, the game animals, the camels, the primates, and the poultry, the domestic poultry, and then jump into human, especially in the market. When a patient gets sick due to acquisition of a virus on the market, they go to see doctors in the hospital, then they spread it to the doctors and nurses, and then they go home and start to spread within the family members. And also, of course, they grow the virus in the laboratory may have problems that leak the virus outside. And of course, all these go into the sewage system of the community. Now, while they jump from the wild animals into the intermediate hosts and then in the market animals, they undergo adaptive genetic changes. The genes change so that they become more easily to infect different kinds of animals and then more adapted to human and finally adapted to transmission from person to person. And that causes a major outbreak. Now, by understanding this process, we are able to set up different levels of surveillance at the wild animals, which I have been doing for quite a long time, and then the animal-human interface, and finally the patient, especially our hospital surveillance. Now, so we have ways, although we don't understand the complete total microbiome in every way in the planet, but we know that there are strategic places where surveillance could be cost effective and perhaps time effective. Now, so these are the lessons from Hong Kong. So this is 1997, start with in March and April, we have a farm chicken outbreak. And then we have the first case of avian influenza H5N1 in a child. They killed the child, actually, the child died. And then we asked for international advice. And the international advice is that, oh, don't worry, this is just an accident. Now, but 8 months later, we have another farm and market chicken outbreak in October and November. And then, bad news, we have a cluster of another 17 cases of avian influenza H5N1, and that lead to the killing of 1.5 million chickens, ducks, and goose in the market. And then the outbreak stops. So we learned a big lesson that the first sign of an emerging epidemic is often the increased animal deaths in farms or wet markets, and then followed by human infection. And that's why surveillance at the animal-human interface at the farms and also the market is actually very important. The first lesson that we learned in Hong Kong in 1997. Now, so we try to make some predictions. If that happened in Hong Kong, it can happen elsewhere. And in 2003, I made this table, that is the food consumption structure in Guangdong province, because we believe that actually the original virus, what we call the goose Guangdong H5N1, is actually from mainland China in Guangdong. Now, so it's important for us to understand the food consumption pattern within mainland China also. Now, you can see that this is in a city, in an urban population in Guangdong. The poultry consumption per year per person in kilogram in 1993 is 12.1 kilogram. And then 10 years later, in 2002, it goes up to 17.6 kilogram. Now, the rural population, they are poorer, and therefore the consumption of poultry is actually lower, but still increasing. Then I asked the question, what would happen in another 20 years' time? What would happen? And what I did is very simple. I get the data from the Hong Kong government about the food consumption structure of the Hong Kong residents. And you can see that in 1973, our poultry consumption in kilogram per person is 9.6 kilogram. Now, 30 years later, in 2003, when I make this chart, it is 20 kilogram, which means that the poultry consumption, just about chickens, goose, etc., ducks, etc., it doubles. Now, if this again happened in mainland China, it means that the number of animals, the poultry, in the farms and also the wet market would double. And that is an excellent situation, excellent condition for the emergence of avian flu in mainland China. Now, so everything you can basically predict by understanding science. So you step by step, you will predict what is going to happen next. And then that is actually come true. 16 years after the first avian influenza H5N1 outbreak in Hong Kong, which actually is followed by multiple small outbreaks of H5N1 in mainland China. But then 16 years later, in 2013, we have the influenza A H7N9 emerging where? In Shanghai, which is the most important economic center of the Yangtze River Delta. Now remember, Hong Kong in 1997 is also a rapidly flourishing economic center of the Pearl River Delta. This is no coincidence. Everything can be basically predicted. Any place which is economically rapidly flourishing with increasing population, increasing consumption of meats, proteins, therefore increasing number of animals in the farms and market, that is the place where emerging infection can come. And in this case, from the avian species, the influenza A H5N1 and influenza A H7N9. Now, this is the migratory flyways or migratory routes of wild birds. And you can see that Hong Kong is over here. And then Shanghai is in this red star. And this is the East Asian migratory bird flyway. And in the north, you can see that it overlaps with the migratory bird flyway of Mid-Asia and also Africa and also Europe. And of course, you know that we have increasing spread of the avian influenza H5N1 from the Guangdong, of course, then spread to the whole of China, and then spread to Middle Asia, and then to Europe and Africa. And now, of course, you can see that our Asian flyway of migratory birds actually overlap a little bit with the American flyway also here. And then the American flyway overlap with the another American flyway here. And that is just not surprising. That in 2024, which is 27 years after our avian influenza H5N1, we now have H5N1 infected cattle in different farms of the United States, at least in Missouri and Texas, and there are outbreaks among the cattle. And the first cattle to human H5N1 case is in Texas, just reported a few days ago. And therefore, it is also not surprising that since 1997, many researchers in our field are predicting that, hey, the avian flu is going to cause a pandemic in human in a short period of time. But now it's already 27 years later, it doesn't happen. But what surprises us is the coronaviruses. Now, so of course, you all know in Hong Kong in 2003, we saw the first case of SARS-CoV-1 in Guanghua Hospital. And this is the chest X-ray of the second patient, which is the brother-in-law of the first patient, which is a healthcare workers. Professor Liu Jianlong is actually a professor of nephrology. So professors are somehow important. We bring the virus to Hong Kong and then to his relative. And we isolated the SARS-CoV-1, the SARS coronavirus 1, from the lung tissue biopsy of his brother-in-law. And that is the first alert of an epidemic. Remember, a sick incoming traveler who is a doctor, a healthcare worker. And that is, of course, very important because with knowing that the SARS-CoV-1 is the cause of SARS, we're able to make diagnostic kits, consider what are the possible antiviral agents, and one day we would have a vaccine. And our team has been able to prove this disease association with SARS-CoV-1 by collecting all the clinical data and also, of course, the viral load change profiles and also the antibody profiles. Next question, of course, is where is the SARS-CoV-1 coming from? And our team, Professor Guan Yi and also Professor Zhang Bujian, they go into the markets in Shenzhen and also in Guangdong, in Guangzhou, to find the animal source. And the reason is also very simple because in the normal blood donors in Hong Kong, we cannot find any antibody evidence of SARS-CoV-1. So if there is no previous antibody response against SARS-CoV-1, it means that the virus has to be coming from an animal. And finally, they found this, the civet, Guotile, is carrying the SARS-CoV-1-like virus, very similar, 99.8% genetic identity. But after knowing this, we have a big argument among us. Now, science is about arguments. We debate daily on the different issues, and we all have different opinions. And of course, some of us are saying that, oh, the civet is a natural reservoir. It can't be others. And I hold a little bit different view. I think that it might not be. Why I think that it is not the natural reservoir of SARS-CoV-1 and go on to do other animal surveillance. The reason is actually very simple. And a lot of this actually is about genetics. And you can see here, this is the electron micrograph of the SARS coronavirus 1. You can see on the surface, there are projections of protein. We call it the spike protein. It is very important protein because it is the place where it's stuck onto our cell membrane. It attach onto our cell membrane and then enter our cells. Without a spike, the virus cannot enter our cells. And very importantly, if this SARS-CoV-1 infect a human or our civet, the civet or human would produce neutralizing antibody against the tip of the spike so that the spike is blocked, so that it can no longer attach to our cells and enter our cells. Now, by analyzing the spike protein, amino acids, and also the nucleotide, we're able to find something that is very useful. Remember, the spike protein consists of 1,255 amino acids, and each amino acid is encoded by the genetic Morse code, the AUGC, and therefore, since 3 nucleotide encode 1 amino acid, it means that it takes 3,765 nucleotides to encode the whole of the spike protein. Now, by analyzing the differences in the nucleotide and therefore the amino acids on the spike of the CVID viruses that we found in 2003 and that of 2004. So you can see here, the upper 3 here. I can't find the cursor suddenly. Where is the cursor? So cursor is gone. It doesn't matter. You can see that year 2003 and in the column, the under year, and then 2004. We find 32 nucleotide mutations. So 32 here, of which the yellow are those associated with changes in amino acids, which means that there are 20 nucleotide changes associated with amino acid change. We call it non-synonymous mutations. And there are 12 nucleotide changes which is not associated with amino acid changes. We call it synonymous mutation. Now, within such a short period, actually just a number of months, there is already 20 change in amino acids, which is more than those neutral mutations, those nucleotide changes not associated with amino acid changes. It means that the virus is under high selective pressure and has to change rapidly. Now then, it is interesting. Why should the CVID, being a natural reservoir, still the virus is not adapted? All the changes should be rather neutral, no change in amino acid. Now, so I have very good reason to believe that the CVID is not the natural reservoir. And this is what we call my eureka moment. Now, of course, other researchers in mainland China are also doing research. And you can see here, it is a summary of the antibody response in the CVID specimen from farms. They have CVID farms, 果子狸養殖場. And also in the wild, the 野生嘅果子狸. We can't find anybody there, a little bit at most. But in the market CVID, which are put in cages, there are a lot of antibodies there. Somehow the cursor, oh, finally I can find this cursor. And you can see the market CVID is full of antibodies. Now that means that this market CVID are not just getting the virus from some other animals. It is not their own. And therefore the spike protein in the CVID SARS-CoV-1 are changing rapidly within just a few months. There is an unidentified animal natural reservoir of SARS-CoV-1, and we need to find it. And therefore, in collaboration with the Agricultural, Fishery and Conservation Department, especially Mr. Sheck, we're able to get a lot of animal specimens in the wild of Hong Kong. And finally, we found that the Chinese fossil bat, the Rhinolophus sinicus, this very interesting animals, is carrying a bad SARS coronavirus, which is rather similar to the CVID and also the human SARS-CoV-1. And up to 39% of these interesting animals is actually having the virus as detected by RT-PCR in the fecal material. And these interesting animals, you can see that why it is called horseshoe bat is because of the thickening of skin around the nostril of the mouth, because the bats, they locate themselves by shouting a high-pitched sound using a method called echolocation. So the sound reflected, they know that there are insects or wasps elsewhere, they catch the insects and also would not fly towards the wall. Their vision is very poor. They mainly is locating by echolocation. And this is a very important finding. I must say that your teachers and seniors are very important, because when I sent this paper out to Nature, it was rejected. Nobody take care about it. And then I go to find Professor Lepsi Choi to help. He is our president at that time, and Lepsi kindly communicated my paper to PNAS, and it was published within around 2 to 3 months. And that makes us the first in the world to say that the Chinese horseshoe bat is the reservoir, the natural reservoir of SARS-related coronaviruses. At that time, we only know that it is related to SARS-CoV-1. It is many, many years later that we find that it is also related to SARS-CoV-2, the cause of COVID. Now, so 2 years after we published this paper in PNAS, we published a review article in the Clinical Microbiology Review. And in the last paragraph of this review article, should we be ready for the reemergence of SARS? We say this: the presence of a large reservoir of SARS-CoV-like viruses in horseshoe bats, together with the culture of eating exotic mammals in southern China, is a time bomb. The possibility of the reemergence of SARS and other novel viruses from animals or laboratories, and therefore the need for preparedness, should not be ignored. Now, so we make this statement, and that formed the basis of our virus surveillance program in wild animals and domestic animals. And so after many years, since 2005 and then onwards, we have discovered more than 60 novel viruses. Actually, now it's more than 80 novel viruses, of which 30 are novel coronaviruses. And you can see that whole list of things that we have discovered by our team members, which are very important in doing all this surveillance work. I just want you to focus on 3. You can see here is the bat coronavirus HU4 and 5. And then, of course, the bat SARS-related coronavirus. And then here, a porcine coronavirus HU15. Now, so just remember these 3 words, and then we go further. In the year 2012, I was in distress because the funding agency told me a bad message. He said, KY, you are finding so many viruses, especially from animals, with no public health significance. Now, that is very worrying, because once they make such a comment, they're going to stop the funding. And I was very worried. I mean, at that time, Hong Kong U does not have a lot of resources for me. Not even now, I must say. And then one morning in 2012, I read New England Journal of Medicine, and I find this paper by Professor Ron Forshare. He said that they isolate a novel coronavirus, which later is named MERS coronavirus, which is Middle East Respiratory Syndrome coronavirus. And he said here, the closest known relatives are bat coronaviruses HKU4 and 5. And I was very happy because they told us in the summary. And this is a virus that we first found, the ancestral viruses, relatives that we first found in 2006. I immediately sent this paper to the funding agency, and my funding is renewed. Very important. And more importantly is Professor Ron Forshare has kindly given this virus to us for further research on MERS-CoV. Now, I just want to emphasize again the importance of animal surveillance. So this is the dead bird surveillance conducted by AFCD. And sometimes there are people who gave me dead birds where I dissect the bird, take out the internal organs, and try to find new viruses. And with this work, we are able to find something actually turned out to be quite important. Now, we now know that anything that flies is very important. The mosquito that flies is the most important animal that kills the most number of humans. They carry malaria, dengue, Japanese encephalitis virus, etc. But bats and birds which are flying is also very important because in the past, we know that there are three families of coronavirus: the alpha coronavirus, the beta coronavirus, and the gamma coronavirus genus. Okay, 3 different genuses. But through the dead bird surveillance, we are able to find an additional genus of virus called the Delta coronavirus. Now, this is a phylogenetic tree. Actually, it's not difficult to look at it. Those who are on the same branch and with the shortest distance together are closest genetically. Now, so of course, the alpha coronaviruses would cluster together. They would cluster together, the alpha, and then the beta all cluster together. The gamma also cluster together on the same branch. So on different branches and then the same branch. And then we have one additional genus, which is the Delta coronavirus. All the Delta coronaviruses are discovered by our team. As you can see here, that there is a porcine coronavirus HKU15, which is found in Hong Kong, and it is very closely related to the sparrow coronavirus HKU17, which is found in sparrows, right? Now, we do not know how important is this finding until many years later. So this is a paper published in Nature in 2021. And why they published this paper is because there are 3 children in Haiti, they become infected with these porcine Delta coronavirus, which is in here. Where is the cursor? Yes, in here and here. And actually, it is coming from our porcine coronavirus HKU15 ancestrally. And we actually find the virus in 2012, which is 9 years earlier. Again, demonstrating that wild animal surveillance is very important in tackling emerging infectious disease and pandemics. Now, of course, finally, all of you know, this is the 1st of January 2020 in South China Morning Post. They say that a SARS-like virus puts Hong Kong on alert. And again, the market, this time in Wuhan, is the epidemic center for this emerging infection. And I just summarized here again, showing that in 1997, H5N1 is in Hong Kong market. 2003, the CVID market in Shenzhen, Dongmen Yesheng Dongme Xichang. 2013, we are the first group in the world in collaboration with Professor Li Lanjun in Zhejiang University. We find the H7N9 in the Zhejiang market. And finally, 2019, Wuhan, Hualan Hoixinseng, COVID-19 in Wuhan, again in the market, showing the strategic importance of surveillance at the human-animal interface. And of course, the vet market is very important. And again, because of the visionary leaders like Professor Lap Chi Choi, who actually signed a contract with the Shenzhen government so that we have the Hong Kong U Shenzhen Hospital. We discovered the first family clusters of SARS-CoV-2 pneumonia there. And again, you can see that from genomic sequencing, the Hong Kong U Shenzhen strain is here, discovered in the 10th of January. We are not the first to discover the virus, but other groups by George Gao and also another group in Wuhan Institute of Virology, they've come first. We come actually third. And again, you can see that phylogenetically, this is clustering with the bad SARS-related coronavirus that are discovered by us and from others. And that makes, again, makes the point that animal surveillance is important and hospital surveillance is equally important, because by doing this work, we show that actually SARS-CoV-2 can be transmitted from person to person and is coming from the bats ultimately. Now, this is a map that I drew by using some data from the web and showing how we consider predicting epidemic outbreaks. Now, of course, you see here that the Pearl River Delta, where Hong Kong is situated, have 2 major outbreaks. One is the 1997 H5N1 outbreaks, and the other is the 2003 SARS-CoV-1. That is important. And then we predict that the H7N9 would come, avian flu, and this is in Yangtze River Delta in Shanghai, where we have demonstrated that it actually come from the wet market, H7N9. So my next prediction is something around Beijing and Tianjin, Bohai area, because it's always the coastal, economically flourishing area. And I turn out to be completely wrong this time. It's actually at the center of mainland China in Wuhan, which is the central traffic hub of China. All the planes land there, all the high-speed railway converge there, even the conventional railway converge onto Wuhan. And it is actually economically highly flourishing at that time. And it is now reasonable to everyone that Wuhan should be the next epidemic center. And of course, this time is SARS-CoV-2. Now, so all these predictions can be wrong, but it's not too far off, I must say. It's still in the mainland, it's still in an economically rapidly flourishing part of China. I've emphasized to you repeatedly about the importance of animals and human surveillance for the pathogen in next pandemic, in sick or high-risk animals and animal-human phase or others. But the problem is that genomic sequencing is great. But out of those that we found, only a few can be cultured. It's not easy to culture the virus. Now, what we can do now is to use organoids. Organoids are balls of organ tissue, which is derived from adult stem cells, from the animals or human. And now what we are doing at Hong Kong U is trying to make all these bad organoids to culture the bad virus, and other animal organoids to culture other animal virus. And then try to see if the virus can grow in the human airway organoids. And if they do, then they have a high potential of jumping from animal to human. And that is actually what we did. This is Professor Hans Clevers, thanks to him, who teach us on organoid technology. And this is a patient's lung. The patient has lung cancer. So you can see the cancer at the center. And of course, surrounding are the normal lung tissue. And we ask the patient to give us some normal lung tissue. From there, we find what we call the adult stem cells. And from the adult stem cell, we differentiate them into what we call these primitive organoids with very poor cilia formation. And then we devise a proximal differentiation medium that differentiates them so that they have a lot of cilia and become much more compact. And they contain all four types of human airway cells: ciliated cells, club cells, goblet cells, and basal cells. And then this is what we did in our laboratory after learning from, the cursor doesn't, good, yes. So you can see that the cilia are beating rapidly, and the central ball of cell debris and mucus are being moved in a unidirectional manner. So that is very similar to the airway. I must say the best that we can have. And of course, this is just a closer look for you. So the cilia are beating with so much cilia, which we believe it is physiologically much more relevant than the primary bronchial epithelial cells that we harvest from the normal patient using a process called bronchoalveolar lavage. So we put it in bronchoscope to get the cells. Very invasive. Nobody wants to do it. But this is much easier because we use patient's lung tissue, which is cut out for treating the cancer. And we can actually turn this into 2D, two-dimensional. This is three-dimensional, but this one is two-dimensional, and you can see a lot of cilia beating. And that is the best way of growing the bad virus in the bad organoids, and then assess the potential for jumping of this virus into human in the human organoids. Yes. Okay, so you have all these devices to help you to determine which virus from animals is going to jump into human. But we need a rapid diagnostic test for the first detection of these animal viruses and excluding the non-human viruses. That is very important. And I collaborate with Professor Terence Lau in Baptist University and make this multiplex nucleic acid amplification system using microfluidic technology. It was already being sold in mainland China in some places, and now also started to be marketed in Hong Kong. And with these, we are able perhaps to catch the first case. And in cases where we can't find anything, there was nothing found, we do next generation sequencing to determine what is the next emerging infectious disease agent. Of course, besides detection kits, diagnostic tests, we need a broad spectrum antiviral. Making an antiviral takes a lot of time, at least 2 years during the COVID-19 pandemic before we have the drugs like Paxlovid and Molnupiravir. But if you can find a broad spectrum antiviral, then that would solve a lot of problems. It would at least decrease the morbidity or mortality of the patients. And in this case, Hindu has been able to find this caspase-6 and also a chemical called ZVID-FMK. And this actually is a chemical that inhibits the caspase-6, which is normally found in our cells governing apoptosis. Now, why this enzyme is so important is because the caspase-6 in our human body can cleave the virus nucleoprotein, actually all the coronavirus nucleoprotein, into fragments. And these fragments would bind to the interferon regulatory free factor free, IRF free, the interferon regulatory factor free, so that it can no longer enter the nucleus. If it cannot enter the nucleus, if these can no longer enter the nucleus, the interferon pathway will not be triggered. All right, the interferon pathway could not be triggered, the virus can multiply. But if we inhibit the caspase-6, the interferon pathway would be intact and the virus cannot grow. And since all coronaviruses exploit the caspase-6, as long as we inhibit the caspase-6, coronavirus cannot replicate. This is a good broad-spectrum antiviral agent for coronaviruses. And of course, patenting and development is underway. Now, Shuofeng is doing another thing, which is again finding a broad-spectrum antiviral which targets on a host protein called the AP2M1. Now, remember the AP2M1 is actually a cargo protein, a trafficking protein that traffics the virus into the cell to the endoplasmic reticulum so that the virus can undergo all the replication process. By using a chemical called ACA, it stops the AP2M1 from trafficking the virus protein. And in fact, almost all the pathogenic human viruses have structured protein that contain this YXX5 motif, which is the target of ACA. And therefore, again, this ACA can be another broad-spectrum antiviral agent. Finally, we have, of course, we need to have a live-attenuated mucosal vaccine platform. And this is Hongling's work. And you can see here, the influenza virus is our vector. We knock out the NS1, which is a virulence gene of the influenza virus. We place it with the receptor binding domain of the spike. And then we use it to immunize the hamsters. And you can see the hamsters, after being challenged with the Omicron virus, those who are not vaccinated with a very high virus titer, while those animals previously vaccinated with our influenza vector vaccine have no more virus titers in both the nasal turbinate and the lung. And remember, our virus only grows well at 33 degrees Celsius. At 39, it grows down rapidly. And this is given by intranasal spray. And you can give, and you can replace this NS1 with any virus protein that is important in entering our respiratory tract. Now, so remember, we need to do virus surveillance in the animals. We need to have diagnostic tests for rapid diagnosis. We need to have broad-spectrum antiviral, and we need to have a universal mucosal vaccine platform. And of course, our vaccine has been approved by the Ministry of Health, and now they call it the National Commission of Health and something, all right? And you can see that it has been undergone phase 1 to 3 clinical trials, and quite useful, at least in the protection of the vaccinated 82% and unvaccinated 55%. It has been used in 6 million provincial cities in early 2023. Within 1 month, 1 million doses has been administered with reasonably good results. Finally, I just want to say that when the next pandemic comes, we will start with multiplex B-cell detection. And also, of course, sometimes next-generation sequencing. And we do real-time detection of pathogen-X by metagenomics to understand how the virus spread. And then we also do rapid multi-layer manual and electronic contact tracing, which would be much less labor-intensive. And we hope that next time, when the pandemic comes, no more city lockdown, no more closure of school and workplace, no more stopping of traffic, 停工、停產、停業、停課, these should not happen again if we prepare well enough. But can this happen? Well, I believe yes, because I am a great proponent of masking, surgical masks. We actually tell everybody to wear a mask in early February, although I was being scolded by WHO and also CDC for proposing something which is not scientifically documented. I believe that if we can make personalized, reusable, self-disinfectable N95 masks with eye protection, it should be good enough to delay the pandemic until effective and safe antiviral drugs and vaccines are available due to diseases causing the next pandemic. In fact, this graph shows you that at one stage in time, Hong Kong is the best in the world, with the lowest number of cases and deaths in the early part when we do not have the Omicron. Because Hong Kong people are very compliant with face masking. I'm talking about 98%. We counted on the streets. 98% of you are wearing masks, even very early on in February in 2020. And if we have this personalized, reusable, self-disinfectable N95 with eye protection, the next pandemic should be okay. We should not be completely paralyzed. Now, I summarize here that, time is almost up. Our duty is to nip the pandemic, the next pandemic, in the bud by tackling all the emerging infections. We need to do animal and human surveillance for novel viruses, all right? We need to do animal and intestinal airway organoid and human organoid to decide which virus is going to jump species barrier. We need rapid multiplex nucleic acid amplification detection followed by next generation sequencing. We need broad spectrum antiviral for early treatment to reduce morbidity and mortality. We need mucosal vaccine platform to stop the infection at the entry site and reduce the virus shedding so that it would not spread. We need conventional surveillance supplemented by big data surveillance with AI and also electronic contact tracing apps, support drug and vaccine supply. So that's all that I want to say today. And I must tell you some things that I believe is very important when I am a medical student. In 1967, the United States Surgeon General said this: it's time to close the books on infectious disease, declare the war against pestilence once, and shift national resources to such chronic problems like cancer and heart diseases. Now, he said this, our teachers say this to us, to me, in the 1976 to 1981, but be careful when you listen to authority. I must say that many a times they are wrong. And so this is an important lesson for me as a medical student. And this is Professor David Todd, my teacher, who teach me internal medicine. And I asked him one day when I'm a house officer at Queen Mary Hospital, Professor Todd, why do you teach infectious disease? All the lectures on infectious disease are taught by you, but you are a hematologist. And he said to me, why? Because nobody in medicine is now interested in teaching infectious disease. I have to take this up because the subject is really very important for our patients. When it spread, many patients died. And what his words are inspiration for me to do infectious disease and microbiology research. So thank you very much for your kind attention. And I must thank my past and present department, academic staff, scientific officers, students,